Well, hello everyone. So it's time for another little lesson in developmental biology, and particularly development of sexual characteristics. Uh, don't imagine that this is going to be particularly spicy. I'm just interested in why do we get different sexes? Why do they look different? What kind of developmental processes do they go through? Like these fish here, there's a male there, there's a bunch of females there. Why do they look different? What has occurred during development to produce these differences? So what I want to do first, though, is I want to address some popular misconceptions about how sexual specification occurs. That way too often there's an overly simplistic approach taken that you can see promulgated, for instance, in attempted laws by Republicans that try to argue for this kind of pattern of development. That it's very straightforward that you inherit either a Y chromosome and an X chromosome or a pair of X chromosomes. If you get a Y chromosome, that triggers hormones, it triggers gonads, it triggers the development of genitals that change the development of the brain in presumably profound ways, that lead in adolescence to the formation of gametes and secondary sex characters, and ultimately to good straight sexual behavior. On the other hand, if you've got an XX, then you get a different set of hormones, and it leads to different gonads, different genitals, different brain, different gametes, different secondary sex characters, and different behavior. And this is like development on rails. We just got this straightforward process going from step 1 to step 2 to step 3 to step 4, one right after the other, with little interaction between them. Now, another problem I want to point out with this particular diagram that I put together, let me emphasize, this diagram is wrong. This is not how it works. But another problem is, notice that I have done something that people often do, which is to gender all of these processes. That we are going to make, for instance, male gonads versus female gonads. And this in many ways is inappropriate because what we're dealing with is tissue in individuals who have inherited either pair of sex chromosomes that responds to factors in the environment. That, for instance, if the hormone is testosterone in males, you could go to a female embryo, an XX embryo, shoot it up with testosterone, and you would get the same response in their tissues that you see in the so-called male embryos. It's all an interactive process over a prolonged period of time to produce the specifics of the system. So just let me emphasize that again. This is not how it works, okay? This is a cartoon diagram that tries to grossly simplify everything to the way a lot of people think about it, which is just that it's specified by what kind of chromosome you get. That's overly simplistic. It's like saying, I'm going to describe to you how a car is built, and first we start with this lump of metal, and then it goes into a factory and it comes out a car. We're missing all the details, all the fun stuff, all the important stuff, and it makes it sound like it's a fairly routine process. Okay, so how does it actually work? Let's see how reproduction actually develops. And this is my first stab at a diagram to show this, that I've added something that wasn't present in the previous diagram. And that is that there are levels of interaction within this mechanism. That, for instance, if you're producing human hormone of testosterone, it will affect the development of the gonads. And one of the things it will have the gonads do is synthesize more testosterone. So you get this wonderful feedback loop that accentuates the differences. And if you don't get testosterone, you don't make those gonadal cells, and you don't produce more testosterone, so you maintain low levels of testosterone. But these interactions occur at all levels of the system. Everywhere you look, you're going to find that there's multi-level interactions, and there's also interactions with other factors. This cartoon, though, does tend to simplify even too much. That, for instance, I ought to have even more arrows. Yeah, let's have arrows from just about every stage to every other stage, from every process to every other process. And I started to throw them in there, and I could have added more, and it would have just gotten hopelessly complicated. So let's not. Let's stop there. Let's just say it's a given that there's huge numbers of interactions within these processes and between these processes to produce the outcome of sexual development. Okay, now simplify it back again. But I do want to point out one thing, that we're dealing with human tissue responding to signals. And one of the key things that happens in the development of sex is that we have a symmetry breaking event. That something triggers one pattern of development that leads to a whole cascade of interactions that tends to skew the pattern of development one way or the other, that tends to make development bimodal. And in that sense, there is a tendency to produce mostly individuals who are simply definable as male versus those that are simply definable as female. I want to emphasize, though, that that's not the only possible outcome, that there's all kinds of different interactions that can occur in this process, that you know this, that, for instance, you can have a Y chromosome that biases an individual to become male, and, for instance, they then develop male gonads, male genitals. Something about their brain is influenced by something to be more masculine. That part is probably the least well understood and least well defined of all of them. They go through adolescence, they produce secondary sexual characteristics, and they produce sperm. But their bias is to prefer sex with members of their same sex, with other males. Is that a defect? I would say no. It's well within the range of what the human tissue should be able to tolerate. We wouldn't consider it abnormal if a female went through this whole process and then decided it was great to have sex with men. Why should we feel that way when a man goes through this whole process and then something in the environment, some factor, influences him to go another way? And that's okay. But here what I'm saying is simply that there are biases that are present, and the biases that occur early tend to skew the distribution of individuals produced by this complex process in one way or another. I also want to mention another factor here, which is often underplayed. There's not just the Y chromosome, that there's also the egg and the X chromosomes and all the autosomes, and most importantly, the cytoplasm of the egg. We don't talk about that much because it's kind of hard to manipulate. So what's going on in human development is the mother produces a very elaborate, very complex, very rich egg, an oocyte, that's then fertilized by the speedy, stripped down, nearly empty of everything but chromosomes sperm. And those contents of the egg cell make an immense contribution to the development of the embryo. So there's all kinds of proteins, there's RNA, there's lipids, there's all kinds of molecules with high energy that are stored in there, and normal development depends very much on those particular processes being present as well. So we shouldn't ignore that. We tend to focus on that Y chromosome because it seems to be unique, but the egg cytoplasm is also important. It's just that it's also given to daughters and sons and anything else that's produced. Okay, now that leads into another factor, though, that's extremely important, and that is the maternal environment. It would be nice if everybody realized, males and females alike, that all of us were first cultured in a little vat that was just soaking in estrogen. that their mothers were all producing female hormones, and that's part of the background that's influencing our development. You don't get to escape estrogen. Doesn't matter whether you're a boy or a girl. You got a good dose of estrogen in those early months of your development. It's also other factors that, for instance, one good example is the hypothesis that what increases the likelihood of being homosexual is the maternal environment. There is some correlational data that suggests that each male child increases the likelihood of homosexuality in subsequent sons by about 33%. Of course, that's not fixed. If you're the fifth son of someone, that doesn't necessarily mean you're gay, which, of course, there's nothing wrong with that, but it just means that there is a predilection to increase the frequency of homosexuality depending on the number of sons you have. The mechanism is not really well understood. The hypothesis is that, for instance, there are Y-linked factors present in the embryo that can be targeted by the mother's immune system. And with each subsequent child, you're getting vaccinated with baby boys each time you have a son. And that's increasing the likelihood that there'll be something that can affect the development of the child in some unknown way. We do not have the slightest idea, but it may be an example of something that is influencing how the human brain responds. And there have been a few molecules identified that mothers may have antibodies to, which are expressed in the brain, but it's a really murky thing and it's very hard to get at right now. I would emphasize as well that there are many other factors that can influence your sexual preference. It's not just what happened to you in the womb, although that can be a factor, but also behavior as a child, experience as a child, experiences in adulthood. All of these things can shape your sexual preferences. Okay, so we've got a couple of factors here. We got Y chromosomes coming in. We've got the egg cytoplasm, which is loaded up with goodies by mom. We've got the maternal environment, which they're soaking in for 9 months. Then there's also, most importantly, the social environment. That these things affect secondary sex characters. It affects human behavior in complex and profound ways. These factors are also conflicting in how they bias it. There are things that favor what we consider culturally feminine behaviors, and there are things that favor what we culturally consider as masculine behaviors. They vary in different environments, but all of these things are tugging on the individual and shaping how they proceed through life, and that all matters. And lastly, I want to mention one other thing that I'll just touch on briefly at the very end, but it may be very, very important, and that is the physical environment. That individuals are receiving all these signals from their own tissues as they develop. They're receiving signals from their mother. They've inherited predilections from their paternal side. In addition, there's all kinds of influences coming in from the environment. Just to name one, think of things like nutrition. Pregnant women are told, here's the things you should eat. You should get a good dose of vitamins. You should take folate. Don't drink. Eat a healthy meal. All that kind of stuff affects the development of the embryo, and it's a mistake to neglect them. There are also factors that can affect the development of sexual characters, which I'll get to a little later. Things like endocrine disruptors. So physical environment also matters as well. I'd also say that in general, being healthy is an important part of the process of development. Embryos would take all kinds of shortcuts if they're desperately starved for nutrients, for instance. Okay, so let me first dispel a few myths that I hear a lot. One is, I've frequently heard people claim that we all start out as female. No, we don't. We really don't. We also don't all start out as male. What we start out at is as something called the indifferent stage. This one kind of annoys me because it, again, feeds into a cultural bias that if you have an embryo and if the embryo lacks a penis, it is therefore female. But as at least half my audience knows very well, the female genitalia are complex and elaborate and interesting in their own right. They are not simply an absence of a penis. And there is an elaborate set of processes to build female genitalia as well as build male genitalia. Okay, the other myth is that female is the default developmental program. And that one's more complex because in some ways it is sort of true in that if you take an embryo and treat it with, for instance, testosterone antagonists, or if you block the production of testosterone in some way, essentially remove that hormonal influence, it will develop with female morphology for the most part. It often won't be complete, however. The embryo may eventually develop into something that's infertile, but many of the female characteristics will be present in it. I should say in her, shouldn't I? But I'm thinking in an abstract sense here. Okay, so there's things like that that can greatly influence how development proceeds. But there are also things, the mother is not passive, the female is not passive, she's actually actively guiding her own development. Okay, I mentioned this, the indifferent stage. This is where we actually start out. That early in development, you are not male, you are not female, you are something ambiguous, something that is identical whether you've inherited a Y chromosome or not. You're in what's called the indifferent stage. And this lasts for quite a while. It ends somewhere around the 8th or 9th week as the gonads start to differentiate. But at this stage, what you find in the embryo, and I know this may not be the most easy to understand diagram, but this is a cross-section through the tail end of a human embryo. What you see up there, there's a kind of a blue oval. That's the spinal cord, just to help orient you. And we're looking at the caudal part of the embryo. You see the gut there, the hindgut, and what you see in gray is a massive mesoderm, just generic mesodermal tissue, often kind of mesenchymal up there as well. And what's illustrated in this diagram is a little flock of red dots. You see those red dots kind of crawling up the yellow gut and heading towards the mesoderm? Those are germ cells. So those are the early cells that will eventually give rise to sperm or egg. And it doesn't matter whether you got an X chromosome or a Y chromosome, this process is going on. Those germ cells are carrying out their migration into the mesoderm, into something called the genital ridge. The genital ridge is a mass of cells that's on top of that blue structure, which is the mesonephros. The mesonephros is a primitive kidney. So I should do a whole video on just kidney development because it's pretty darn cool. But the kidney develops in 3 stages. Initially, it makes a pronephros, which is a very crude, primitive sort of kidney. Then it makes a mesonephros, which has a lot of the filtration ability. And then it finally will build the metanephros. All the previous kidneys just degenerate and go away. They are vestigial. They are not used for anything. And it's just the metanephros that differentiates into the standard adult kidney and carries on all the functions of blood filtration and so forth. But anyway, this stuff is all happening early when you don't have a metanephros yet, but you have a mesonephros. And these mesodermal cells and these germ cells pile up in a structure called the genital ridge. It's just a mass of cells that's going to give rise eventually to the gonads. Now, in this diagram, you might also notice there's no genitalia. The gonadal tissue is relatively undifferentiated, and it's identical whether you've got an X chromosome, pair of X chromosomes, or a Y chromosome. It's just in the process of building. So that's the indifferent stage. Another key thing about this early pattern of development is that there are ducts. Yeah, sex is mainly a matter of all kinds of tubes organized in specific ways. And the two tubes we care about here are the mesonephric duct and the paramesonephric duct. The mesonephric duct is often called the Wolffian duct, but nowadays we tend to avoid naming things after old scientists, so we now call it the mesonephric duct. And the paramesonephric duct is sometimes called the Mullerian duct. Again, we've changed the name for the same reason. Okay, so what you see here is in blue, there's a tube. That's the mesonephric duct. And what that does is it is the duct that connects that primitive mesonephros, that kidney, to the cloaca, or the urogenital sinus as it's called here. So this is part of the filtration system. It's in some ways it's kind of like the urethra in an adult. So it's got the same role as it's carrying waste products out to the cloaca for disposal. But it is fully hooked up into this primitive kidney. It contains these things called mesonephric tubules, which terminate in structures called Bowman's capsules and glomeruli. If you know your kidney anatomy and kidney function, you know what those are. Those are actually the filtration units of the kidney. So it is acting as a filter here. In addition, there is something called the paramesonephric duct, which develops in parallel with the mesonephric duct. It seems to be induced by the presence of the mesonephric duct. So the mesonephric duct is sending out cell-cell interactions, it's sending out signals and communicating to its neighborhood, and it's inducing the formation of this tube that more or less parallels it. On one end, it does terminate in the cloaca, so that looks kind of familiar, like that's just like the mesonephric duct. But the other end is just a blind opening. It just opens up into the coelom of the embryo. It does not have any filtration functions. It's associated with the mesonephros, but it's not working with the mesonephros. It's just sitting there like a pipe waiting for something to be done with it. Okay, so let's look at these organs as they develop. So this is the testis. On the left, what you see is an early diagram of it. Again, you see that mesonephric tubule. So that's that kidney function that's over there. And at this stage, at the 8th week, it's degenerating. It's just falling apart. So we're losing that Bowman's capsule. We're losing the glomeruli. We're keeping little bits of the ducts, but that's about it. The whole structure is now differentiating in a new way. It's making these things called testis cords. And this is where the various tubules are all going to connect. There's going to be tissue in here that's going to differentiate into sperm, for instance. It will transport down these ducts into the mesonephric duct, which will become the vas deferens and things like that. And then the gametes can pass out of the individual to some greater cause. Okay. Notice here, in blue, we have the mesonephric ducts, but we also have in red the paramesonephric duct. It's still there. It's not doing anything yet. How strange. It's just, again, still just it's just a tube that just ends there, and it's not really connected to anything except the space in the coelom and the cloaca. So it seems kind of useless. Now, what's also going on here is this structure is induced by the presence of testosterone. And in response to testosterone, it produces more testosterone. So we're going to make lots and lots of testosterone with this thing and sort of saturate big regions of the embryo with that hormonal signal. Another signal is called AMH. This is it's got a nice name. It's the anti-Mullerian hormone. Remember that paramesonephric duct, which also is called the Mullerian duct? This is a hormone that actively induces the paramesonephric duct to go away. So it's a signal that makes the paramesonephric duct dissolve over time. And also what it's producing a reductase enzyme. well, actually, other tissues are producing the reductase enzyme. The testosterone is going to go through the embryonic bloodstream, and it's going to arrive at places like the prospective genital region, where the cells there convert it with this enzyme into dihydrotestosterone. Dihydrotestosterone then induces the genital tissue to develop into things like penises and scrotums and so forth. So that is the side of what we see when the embryo inherits a Y chromosome. It's going to do all that stuff. It's going to make a testis. It's going to adapt and use the mesonephric duct, and it's going to destroy the paramesonephric duct. What about in individuals that do not have a Y chromosome, that don't have a lot of testosterone around? What you see on the left is, it should remind you of the previous diagram. There's a degenerating mesonephric tubule in blue. That's the mesonephros in gray. And again, it's degenerating. We're losing the Bowman's capsule. The ducts are kind of retracting. And we're also seeing now the formation of gonad. There are germ cells migrating into it, and the developing tissue is maturing in specific ways. And on the right, what you see is what it will eventually end up looking like in the 5th month. This is different from a testis. This is an ovary, and it contains oocytes, lots of oocytes. The oocytes are big cells. They're surrounded by follicular cells. And what's eventually going to happen to them at around puberty, this will start happening, is that one at a time, these will mature, and then they will erupt and spew the oocyte into the coelomic cavity, so the space around the gonad. This is different from what the testis does. Remember, the testis is also making gametes, but it's dumping them into that blue mesonephric duct to head on out. The ovary is dumping them out into the surface, and that sort of makes sense because oocytes are big and they probably would not go neatly through these efferent ducts. So what will happen is those efferent ducts and the mesonephric duct will be useless, and they'll eventually degenerate and go away as well. You might be wondering what happens to that poor oocyte that's just been spewed out of the gonad. What it will do is find its way to that open tube, the paramesonephric duct. In the adult, this is going to become the fallopian tubes. So it'll find its way there, and then it will enter that tube and go down to where it needs to go to do its job. You will notice in this diagram they're not particularly close to one another, and they aren't. There's going to have to be some rearrangement of the tissue to get it to the right place. Okay, what kind of signals are at work here? Estrogen is the important one. You could say that one signal is the absence of testosterone. That's also a factor. As I mentioned, the maternal side of things is the one that's carrying the XX chromosome is not necessarily passive. It's also actively working. So there is also a hormone called this is a real mouthful. It's called the chicken ovalbumin upstream transcription factor 2, or COUP-TFII for short. So it's going the gonad is going to produce this hormone, which acts like AMH did for the testis. It will induce the degeneration of, in this case, the blue mesonephric duct. So we'll get rid of that. And that requires a production of this hormone in order to do that. If you don't have, well, it's actually a transcription factor. If you lack this transcription factor, the mesonephric duct will hang around forever, and it will actually exist and differentiate into some other tissues, which isn't necessarily good for the individual. Okay, so we've got a couple of gonads in the works. This is kind of a summary. If you have testosterone, if you contain testosterone, these two, they look just the same, right? Pretty much the same. If you have testosterone, the blue ducts will persist, the mesonephric duct will persist, the gonad will develop into a testis, And the red duct, the paramesonephric duct, will be obliterated by the secretion of AMH. On the other hand, if there is no testosterone in the environment, you get the situation on the right, where you have degeneration of the blue duct. The mesonephros is going to go away under the influence of COUP-TFII, and you're going to differentiate an ovary rather than a gonad, or rather than a testis, and the paramesonephric duct will persist and differentiate into some other tissues that I'll show you in a moment. So all this is an illustration of the sixth week when they look pretty similar, and it's just the difference in testosterone is going to lead to different fates for these tissues. So just a little bit more about the female reproductive system, the ovary. In the absence of testosterone, you're going to make this. So on the left, you see the earlier stage, and on the right is what's going to happen as the individual matures. That ovary has to get someplace a little closer to the fallopian tubes, okay? So there will be migration of the ovary towards the ends of that paramesonephric tubule, which is now called the fallopian tube. And even there, it's kind of a jury-rigged situation, because it's still going to just be popping out oocytes into the coelom, and it's just going to drift into that open tube. This is why you sometimes get ectopic pregnancies, is if an oocyte emerges, doesn't make it into the fallopian tubes, it can plant somewhere else and develop. Okay, so there's the fallopian tubes in red. The ends of the paramesonephric duct develop into some rather prominent structures. They develop into the uterus, for instance, and they develop into the upper portion of the vagina. So the paramesonephric duct, which is completely eliminated almost in males, is going to become these rather important structures in the reproduction for individuals who are XX. Also of note here, note there's a couple of structures in blue in that adult reproductive system. There's something called the epo-ophoron, and there's something called the para-ophoron. Those are the pitiful, feeble, decrepit remnants of the good old mesonephric duct. It's not totally gone. It's still lingering there. These are visible in these mesenteries and the ligaments around the ovary, but they don't do anything. They're just vestiges of the old system. Okay, so let's summarize what happens to these things. Under the influence of testosterone, the mesonephric duct and the paramesonephric duct have different fates. So if you have testosterone around, the mesonephric duct will develop into things like the vas deferens and the epididymis. It's part of the transport system for getting sperm from the testicles to the end of the penis. Indirectly, not directly by testosterone, we also get the differentiation of things like the penis and the scrotum and the prostate by dihydrotestosterone. Okay, if testosterone is around, what happens to the paramesonephric duct? It becomes vestigial too. It forms a couple of very, very obscure anatomical structures. One is the prostatic utricle, which is simply a short dead-end tube around in the prostate near the ducts of the vas deferens. And it also makes something called the appendix testis, which is simply a little bump in the testis near the epididymis. That's about it. So again, it's totally useless stuff. Okay, what if there is no testosterone around? If there's no testosterone around, the mesonephric duct is going to go away. It's going to make that thing called the epo-ophoron and the para-ophoron, both vestigial, just both little traces left in a little bit of membrane there. But more importantly, the paramesonephric duct will expand. It will form the uterine tube, the uterus, the upper part of the vagina, And in addition, you're going to have the production of estrogen, which will induce the development of the clitoris, labia, and the lower part of the vagina. You don't want to have just the upper part. You want both parts together, or it doesn't work. So it's going to differentiate all those tissues. One important thing I want to mention here is that these are all interlocking, interlaced components of the developmental system, but they're also somewhat independent. So, for example, you can have disorders that cause a failure of the female genitalia to develop, and they still make the uterine tube, the uterus, and the upper part of the vagina. It's just that the lower part is not connected, and that causes some serious problems, of course, if the individual wants to have children. Similarly, in males, you can have testosterone that induces the formation of testis, vas deferens, epididymis, all this kind of cool stuff. But there's something interfering with the production of DHT, for instance, and then the external genitalia fail to form properly. So all of these are kind of interdependent and all working together, but at the same time, there are ways to dismantle and take apart components of this process. Okay, let me go back to this diagram I showed you earlier. That here we've got all these interactions going on, and I've told you already that those bottommost three, the formation of the secretion of hormones, the production of human gonads, the production of human genitalia, are all dependent on external factors in the environment. The Y chromosome is a biasing component. It tends to switch production to the male pathway, but it doesn't always work. Usually it works, but not always. But even with that, all these other parts are independent, interlinked. They are cell non-autonomous, if you know what that means. That means basically that they are dependent on external factors to carry out the proper pattern of development. And you can get some complicated variations on the pattern. Then they're all perfectly normal responses of perfectly normal human tissue to conflicting signals or different signals or changes in the signals. It happens all the time. Okay. So the important lesson here is that human reproductive tissue has complex plastic responses to their history and to interactions with other tissues. Even the earliest decisions are not absolutely defined by something as simple as a Y chromosome. Don't make that mistake. Y chromosome is a biasing factor, not a defining factor. We regulate and adjust our cell biology all the time to suit the circumstances, and that's kind of a cool property to have. To be plastic and flexible is a good trait for an organism to possess. But okay, just to close this, I want to say a little bit about those green arrows because I really haven't talked about them much. This last semester I taught a course in ecological developmental biology. It was a blast. I learned a lot. I hope the students learned even half as much as I did. It was kind of cool. So apparently I have the ability to talk about this subject for like 3 months. I won't, not today. But it's a subject that we could come back to at some later point. Just let me know if you want to hear more about it. So I'm just going to bring up one relevant point about the effects of the physical environment on sex organ development, and that's the effect of endocrine disruptors, which you may have heard of. So here's a couple of nice graphs that should scare you a little bit. So what we're looking at there is if we start at in A, top left corner, that's a graph of the age-adjusted incidence per 100,000 or 10,000 population of testicular cancer. Notice the line is not going down, it's going up. Year by year, this is increasing frequency of testicular cancer. Also note that this is the age-adjusted incidence, so you can't just simply say it's because we live longer, so therefore we're getting an increased frequency of these problems. It's actually been compensated for that. So this is saying there's a clear trend, an upward increasing trend in the frequency of testicular cancer. Note top right, there's another graph, similar graph for breast cancer. And breast cancer has also been going up. We do have to say that this is the initial incidence of these diseases, and we're getting better and better at treating them. So if we were plotting, for instance, death from these diseases, that would be a different story. But we're just asking, how often does this come up? And it's surprisingly often, and it's increasing over the years. Over the last few decades, it's been going up and up. The lower left is also an interesting medical problem. This is the frequency of hypospadias. So in hypospadia, the penis is a tube, right? It's a hollow tube. It's got a urethra running up the middle of it. And during development, it forms by like a hot dog bun, kind of closing around and then sealing. And if there's a failure in that, the tube does not close completely, that what you may have is an opening at the end for the urethra, but also an opening at the base where urine and semen can dribble out instead of going all the way to the end. It's not something you want to have happen to your children, okay? But nowadays it can be treated effectively pretty quickly. It just takes a little surgical work. Stitch it closed and everything is fine once again. It is a delicate tissue though, so you really don't want it to happen. But look at there, hypospadias have also been going up year by year. This is a birth defect. Officially, it really is a birth defect. It's a manageable one, but it's still a birth defect. And the rate of those birth defects have been climbing and climbing over the years. All right, so the last graph, the bottom right, this has got to be a relief, right? Because there, the lines are going down. Yay, it's not going up, except that what's being plotted here is sperm count. And that's kind of impressive, that sperm counts have been declining over the decades. Look at that, from about 1930 to about 1990 in America, sperm counts dropped by about half. Compare Europe in 1960 to the United States. Man, they were manly men back there in Europe then. But their sperm counts have been declining as well, and now they're very similar to ours. This is a worrisome trend, okay? It's nothing to panic about just yet, but it may mean that it's more trouble to impregnate somebody. That there's going to be a reduced fertility. There's a cost to this in our behavior and in our lives. Is it a terrible thing? Not yet. I would say, after all, I have 3 children, and so all I really need was 3 sperm total, if they could be efficiently delivered. But no, this is going to affect fertility and is something we ought to consider for the future. And we ought to be asking ourselves, why are these things happening? Why are we seeing an increase in birth defects and cancer and a decrease in sperm counts? And the answer seems to be environmental factors that are basically poisoning us at a low rate. One example that we're beginning to get a handle on is that, for instance, that sperm count stuff, one of the things that affects that is phthalates. Phthalates are a really common chemical that is used in, for instance, hardening and curing plastics. Are you surrounded by plastics? We are. There's lots of plastics around. You have probably smelled phthalates. This is the source of that new car smell. Yeah, so get a new car, sit in there, take a deep breath. You may like that new car smell, but just imagine, oh, I'm killing sperm every time I take a breath here. So that's one concern, is that things like phthalates. Phthalates are also used in fracking chemicals. Lots of places where phthalates are commonly used in industrial processes. And what we consider to be likely is that there's an increased release of these kinds of industrial chemicals of various sorts, not just the phthalates, and the phthalates aren't the only thing, so you can't just blame those. But we've got an increasing release of these chemicals into the environment, and they are having serious effects on our reproductive systems. We ought to do something about it. I don't know what, but I'll let you think about it. Okay, so I'll just end there. I've been babbling long enough. If you want to hear more about endocrine disruptors or environmental effects or anything like that, drop me a note and I'd be happy to do something about that. I got to think about what I'll do next week. And for now, let me just close with some credits here. My name is PZ Myers. I write and blog at freethoughtblogs.com. There's my URL up there at the top, freethoughtblogs.com/pharyngula. And I also write there with a large collection of names. Look at all those people on the left. Those are all blogs that are there at Freethought Blogs. Lots of good stuff going on there. Please do stop by and have a conversation with us. And with that, I'll let you all go, and I'll think about doing something for next week.